name is Christina, and today we'll be talking about B lymphocytes. B lymphocytes can also be known as B cells. They are a crucial part of the immune response. They develop from stem cells in the bone marrow and mature in the secondary lymphoid tissue. The B cells are in charge of producing a protein that we know as antibodies. They target specific antigens in pathogens or other foreign substances such as toxins. If it weren't for the B cells, we would not be able to fight any pathogens. B cells have a very unique structure with a marginal zone that engages with signaling pathways for development and retention. B cells have a complex structure with many immunoglobulin receptors that are crucial when it comes to immune regulation and recognition of antigens. B cells are very significant when it comes to diagnosing chronic lymphatic leukemia known as CLL. This diagnosis is performed by looking at the B cells' phenotype and count. These results will also help distinguish between B cell lymphocytosis and CLL.
When a pathogen is present in the body, the antigens attached to the surface alert the body's immune system. They serve as markers that tell the body that there is a foreign substance that needs to be eliminated. B cells have receptors that will bind to the antigen. The antigen will then be broken up and presented by an MHC2 protein on the surface of the B cell. When helper T cells bind to this MHC2 complex, it activates the helper T cell, which is important for B cell proliferation. These activated T cells will then bind to B cells, allowing the cells to differentiate as antibody-producing plasma cells and memory B cells. The antibodies produced are specific in the destruction of the antigen. Some ways that antibodies can inhibit the pathogens are through neutralization, opsonization, and activating the complement system. Memory cells are able to remember the specific antigen, so when the body gets exposed to the same pathogen in the future, our body is able to quickly defend against infection. Our humoral immunity is important because it helps our bodies fight against pathogens which can cause diseases. Vaccinations are also an important example of humoral immunity because it exposes our immune system to these antigens, creating memory B cells that will quickly recognize antigens in the future.
Hi, my name is Anna and I'll be talking about T lymphocytes. T lymphocytes are developed in stem cells in the bone marrow and mature in the thymus. They play a critical role in recognizing and responding to pathogens. For the structure, all lymphocytes have the TCR and the CD3 receptor. The TCR is a transmembrane receptor made up of various alpha and beta chains. The CD4 co-receptor is found on helper T cells and they bind MHC class II molecules. CD8 co-receptor is found on cytotoxic T cells and they bind MHC class I molecules. There are also many structural markers that depend on their type and role. For their functions, the cytotoxic T cells destroy the infected cells either through the FAS ligand pathway or the release of perforins and granzymes to induce apoptosis in infected cells. The helper T cells are responsible for carrying out cell-mediated immunity. At first, they are inactive and need to be triggered to carry out immune responses. This activation happens when they interact with specialized antigen-presenting cells, mainly dendritic cells in the lymph nodes. Lastly, T regulatory cells suppress overactive immune responses, ensuring balance. For the clinical significance, when T cells are low, that may indicate a condition like HIV. Meanwhile, when T cells are high, that could be the result of certain cancers such as T-ALL or ATL.
Hello, my name is Zach Hargrave, and I am going to be briefly explaining how our immune system achieves immunity using T lymphocytes. When pathogens invade the body, immune cells like macrophages and dendritic cells engulf and destroy them. After this, they display the pieces of the pathogens they just destroyed, known as antigens, on their surfaces, signaling the need for an immune response. Then, T cells, specifically CD4+ helper T cells and CD8+ cytotoxic T cells, recognize the antigens through their receptors. The CD4+ T cells play a crucial role in activating other immune cells, while the CD8+ T cells are tasked with directly killing infected cells. Once activated, T cells undergo proliferation, rapidly multiplying to create many identical cells. At this time, they also differentiate into effector cells that carry out specific functions. The CD4+ T cells produce cytokines that further stimulate the immune response, and the CD8 plus T cells prepare to attack and eliminate infected or cancerous cells. CD8 plus T cells also release molecules like perforin and gran enzymes, which helps cause the destruction of infected cells. And CD4 plus T cells enhance immunity by activating B cells, which are what produce antibodies to neutralize pathogens. And the antibodies also boost macrophage activity. After the immune response, some T cells become memory T cells, which remain in the body for a long time. These memory cells enable rapid and effective response if the same pathogen is encountered again in the body. This intricate process showcases how our immune system not only protects us, but also adapts to future threats. Thank you.
For this slide, I'll be discussing the nature of antibodies and the crucial role they play in our immune system. Antibodies are specialized proteins produced by our immune system. They protect us when an unwanted substance like pathogens enter our body. By binding to these invaders, antibodies help eliminate them. They also play an important role identifying and neutralizing pathogens like bacteria or viruses. It provides both immediate defense and long-term protection against infections. Antibodies, also called immunoglobulins, have a Y-shaped structure that consists of 4 polypeptides, which include 2 heavy and 2 light chains. There are 5 main types of antibodies with each specific function. Antibodies have numerous applications in medicine and research, along with 5 key main functions. Overall, antibodies are essential components for our immune system, playing an important role in our health and protection against diseases that enter.
The clonal selection theory explains the adaptability of the immune system by using its pre-existing lymphocytes. It has the capability of producing B cells and T cells that are capable of responding to specific antigens that enter the body. The lymphocytes are each programmed to the specific antigen that enters the body in order to be able to help it fight off from infection. This concept can be easily explained by using the B cells specifically, since they are constantly being made by the lymphoid stem cells. Each B cell is made to produce a specific antibody against a specific antigen that is entering the body. This can be referred to as the antigen of its dreams, since they are both basically made for each other. And this is only for B cells with a specific receptor that can be activated by the antigen. So 1 cell is equal to 1 antibody because they specifically code for each other, but more specifically the B cell codes for that antigen. In order to do this though, it needs help from the T helper cells, which signal its activation. The T helper cells are activated when a macrophage breaks down the antigen, and then it just becomes sort of like these pieces that then the T helper cell can recognize, and then it can be used in order to attach the T receptor onto the B cells class 2 MHC. This is what activates the B cell specifically, which is why it needs the T helper cells in order to activate properly. Once the B cells have been activated, they then are able to start multiplying. This is a really big part of the whole clonal selection, as they turn into plasma cells, or they can turn into memory B cells. The plasma cells are responsible for producing the actual antibodies for the specific antigen, and then the memory B cells remain in circulation, but they're just there for future production, so that if the antigen were to come back again, then it would be able to properly have a secondary response that's more effective in getting rid of the antigen. And this whole process is basically to help against infection in the body. The antibodies are the actual pieces of this whole process of clonal selection They're kind of like the goal that the whole process goes through all of this work to get to, because the antibodies result in the creation of basically something that's able to neutralize foreign invaders and then have them destroyed. This is what actually prevents the body from infection, and that's why this process is so important.
Hi, my name is Jocelyn, and I'll be covering the expansion of lymphocytes. Lymphocyte expansion is also referred to as clonal expansion. It serves as rapid cell division of identical cells from a single parent cell. It increases the number of specific lymphocytes as a protective response from the immune system against pathogens inside the body. The steps toward the expansion of lymphocytes includes antigen recognition. This is where B and T cells contain specific receptors that recognize and bind to specific antigens. The second is activation. This is where the binding of the receptor to the antigen sends signals inside the lymphocyte, causing it to activate. The third is clonal expansion. This is where the lymphocyte starts to multiply rapidly, producing identical copies of itself to target the specific antigen. And the fourth is response. This is where clones either become effector cells or memory cells that fight off the specific antigen. Thank you.
Hi there, this is Livy, and I'm going to be doing the Christian worldview perspective for this unit. The verse that I found that I thought fit this unit was Jeremiah 33:6, and it says, "Behold, I will bring to a health and healing, I will heal them and reveal to them abundance of prosperity and security." So what this verse is saying in regards to microbiology is that God is our ultimate creator, healer, and provider. God is our creator because he created us with a defense mechanism, our immune system. And he provided us with cells to defend us against different pathogens and dangerous substances to keep us safe. And then also he is our healer. He has the power to heal people who are sick or injured or are fighting infection or some disease or disorder. And he also uses that time to provide us with an opportunity to trust him more and to deepen our faith in him. And then additionally, God is our provider. And he provided us with the ultimate sacrifice, his son, to pay for our ultimate sickness, which is sin. And he gave us the option of choosing to acknowledge that and to follow him, or to stay in our sickness. And then last but not least, while microorganisms and cells fight in our bodies to keep us safe and to keep us from harm, God is the one who is ultimately in control of our life, and that includes our health and the way we live. Thank you.